to have you here today. Dr. Bharti, tell us a little bit about your laboratory and the work that you've been doing at the NEI.
So yes, thank you very much for having me. My name is Kapil Bharti. I'm a tenure track investigator at the National Eye Institute, and my lab works on stem cells and their applications, translational or clinical applications for ocular diseases. And there are different parts of the eye. They focus on some fundamental aspects of how stem cells can be used to better understand ocular diseases or even ocular development. But then we translate those findings into clinical applications where we want to develop new cell therapies for patients with macular degeneration and also understand how actually macular degeneration initiates and progresses on to these advanced stages so that we can have a possibility of intervening in the disease at a very early stage with drugs or molecules or small molecules or biomolecules and pursue that clinically.
Right. So this time around, you're here to talk about a very exciting living model that you've created. Tell us about the model and how inspiration came about for you.
So as you're aware, age-related macular degeneration is a disease where patients go blind, and blindness happens because the photoreceptors die. And we all know that photoreceptors are the key cells that are needed for seeing light or vision. And the reason photoreceptors die in these patients is because the pigment epithelium underneath the photoreceptor starts to die off. But when you look at some of the anatomical analysis done on patients' cadaver eyes, you notice that not only the pigment epithelium is dying, it's also the choroidal blood supply that's dying in these patients. And that's the dry form of AMD. But there's the other advanced stage, the wet form, where the choriocapillaries do the opposite. They penetrate the RPE monolayer, the pigment epithelium monolayer, and leak fluid and blood in the subretinal space, again leading to blindness because the photoreceptors are affected. So we figured the common element that in both these advanced stages is the pigment epithelium and the choroidal blood supply. So if you really want to understand the disease, how it initiates and how it progresses, you want to not only look at the pigment epithelium, you want to also look at the choroidal blood supply. So hence, that's the motivation for us to kind of put the two tissues together, and we wanted to do it from human cells. So hence the need for human stem cells and create a three-dimensional living tissue of pigment epithelium and choroid made from AMD patient-specific stem cells in a dish, so we can actually recreate these two diseases in the dish and study its progression.
Can you describe the model to us?
So the way it works is that there's a technology called iPS cells, or induced pluripotent stem cells, where you can start with the patient's fibroblast or blood cells, reprogram them to become this iPS cells. And these iPS cells, for all practical purposes, are identical to ES cells. But the advantage is you can make them from patients on somatic cells, blood or fibroblasts. And then we can take those iPS cells and differentiate them into the pigment epithelium monolayer or the endothelial cells. And that's what my lab does. So we have optimized protocols how to make these iPS cells into pigment epithelium, the endothelial cells, and then we use a very interesting new technology called 3D bioprinting, which is very similar to 3D printing that now is used everywhere for making plastic molds. But 3D bioprinting focuses on cells, and you can essentially print cells in any order you like. So what we do is we take the endothelial cells that we took from AMD patients' iPS cells, mix them with a hydrogel, and make kind of a paste out of it, which we call bioink, just like ink used in printers. And then we print blood vessel pattern. And these cells, as they should, they start to recreate vessel-like pattern on that printed structure. And that creates for us, because it's mixed with fibroblast and other cells of the choroid, it creates for us the choroid of the patient. And then these cells are on a scaffold, so we flip the scaffold upside down, and on the other side, we make the monolayer of pigment epithelium. We let the tissue mature for 4 to 5 weeks. And as the tissue matures, the two cell types start interacting, start talking to each other. And in fact, we have seen in our culture models that the pigment epithelium starts to control the growth of blood vessels in this dish. And that's the whole model. And so we think we have created a three-dimensional back of the eye in a dish.
When you say control, What elements of control have you seen?
So, for the blood vessels to grow, for endothelial cells to form blood vessels, they need a growth factor called vascular endothelial growth factor, or VEGF, and we have to add that in our culture condition, in our culture medium. Once the vessels are formed and RPE monolayer is formed on the other side, we actually remove VEGF from culture medium and let the vessels be dependent on whatever VEGF RPE is secreting. And we've seen that RPE secretes sufficient amount of VEGF that the vessels keep proliferating and keep staying maintained. And if we actually remove the RPE and do not add VEGF from outside, the vessels die off. That's how they're controlling the VEGF growth, the vessel growth. In fact, this is what also happens in dry AMD. And we're able to demonstrate the same phenomena in a dish. And now we did the opposite experiment where we stressed the RPE cells, and that caused them to secrete almost fourfold higher levels of VEGF and other inflammatory cytokines like IL-6, interleukin-6, and interleukin-8, which have been implicated in inflammation and AMD in the back of the eye. And lo and behold, these vessels start to hyperproliferate. They migrate up towards the RPE, and we've now seen in some cases a couple of vessels just penetrate through the RPE monolayer, much like what happens in wet AMD. So that's why we know that it is the RPE which is a central component controlling the vessel growth, their proliferation, and maintenance.
That's very exciting stuff. Tell us about perhaps some of the next level that you're going to explore in terms of signaling and control.
So, for us, we're excited to have this kind of a Lego box in our hand, and now it allows us to, first of all, see what these cells are, how these cells are talking to each other. How does the RPE talk to the choriocapillaries, and what are the really initiating events of disease by gene expression level and the signaling level, so that we can come up with drugs that can intervene at those levels and stop even disease from initiating at a very early stage, or even delay its progression in patients where it has already initiated. And that's one aspect of it, where you can really study disease progression and its disease-causing pathways. But then the other aspect is, as you know, AMD is a complex multigenic disease. There are many risk factors have been linked to AMD, but none of them has really been connected to any pathological findings really at the cellular level. Because we can make iPS cells from patients, this gives us an opportunity to really make iPS cells from patients with different genetic backgrounds, make their tissue in a dish, and see if the tissues behave differently, if the disease happens differently depending on the different genetics of the patients. So not for the first time, we have the opportunity of really linking the genetics to cell biology to disease pathways from using patient cells. And the third thing is, we can take this whole tissue, if we make it from a healthy person, or even from the same person, because AMD is a slow progressing disease, and convert it into a transplantable tissue. We can transplant it back in patients who have lost the pigment epithelium or the choroid along with it, and rescue the pigment epithelium and the choroid. So all three opportunities are there.
What about using this as an in vitro model for the testing of therapeutics?
Yes, absolutely. I think that's that's the first aspect. Where you uncover disease-causing pathways, and then you're able to test different drugs. So we're working in close collaboration with the NIH Center for Drug Testing, which NCAT, Center for Advanced Translational Sciences, that have the possibility of testing several thousand drugs in a robotic fashion. So the one thing that we're now working on is taking these models and miniaturizing them so that we can fit them in multi-well plates, so that we can test several thousand drugs and see which of the drugs would inhibit disease-initiating events in these cells.
What lead therapeutic candidates are you looking at?
So in this case, we're going for a cellular phenotype. As you know, one of the initiating events for AMD is drusen formation. Since we have been able to recreate drusen-like particles in the system, we're going after which drugs would inhibit or block drusen formation. And in a way, and that's the advantage of doing this large-scale drug screens. You don't have to be biased towards a drug candidate. You say, I want to go after this clinical phenotype. Any drug that would inhibit or that would suppress that clinical phenotype, that's my favorite drug. So we're going to go for an unbiased screen And usually those libraries include, kinase inhibitors, epigenetic modifiers, inhibitor of transcription factor pathways, and that would include already, a few thousand drugs, just doing a calculation in my mind. And I think that's the opportunity, because you can just go unbiased and pick out the 3 drugs that give you the strongest phenotype.
So tell us what's on the menu for the next year with this model, in the short term and then in the long term.
So in the short term is what we just discussed. We're going to be going after disease pathways and really convert this into a screenable assay so that we can start the drug screens as soon as possible. And in the long term, we want to really convert it into a clinical grade transplantable material. You might be already aware that there's a number of different RPE-based clinical trial, transplantation of RPE-based trials going on using ES cells. At NEI, we're focusing on a trial for autologous iPS-derived RPE for patients with geographic atrophy for the dry form. So that's already underway. We hope to start the trial next year in 2018. But that's in that trial, we're only going to be transplanting the monolayer patch of RPE on a scaffold. We won't have the choroid. But the next step for us is to actually work on a trial which would also have the choroid. Because as I said early on, even the chorioCapillary is missing in advanced patients. So we want to supply both of them to these patients. But that is a slow process. Converting this 3-dimensional tissue into a clinical grade, GMP grade tissue takes a few years to develop. And then to do all the preclinical testing in animals where you demonstrate safety and efficacy of your product, that's going to take 5 to 6 years. So that's what's next for us.
And when we look more closely at the interface between the choroid and the RPE, are you actually replicating a true blood-brain barrier with all of its transport controls and mechanisms?
So from what we can tell from all the experiments we have done, it looks like. Whether how true it is, I would find out very soon, because for me, true is how close it is to a native condition, and I would not just say, yeah, it looks like that. I would do at least 6 different experiments and say, does it replicate this finding in terms of barrier permeability? Does it cause polarized secretion of cytokines? And does it cause polarized vectorial transport of water from one side to the other? So all those assays are the properties of the blood-retina barrier. And we're in the process of doing those experiments right now. So I will tell you very soon that how true it is.
Terrific. Dr. Bharti, tell us a little bit about some of the other things you're doing in your laboratory.
So my lab really focuses on harnessing the power of the iPSL technology, because we think this technology really has revolutionized how we do science and how we look at really the patient material. So we have, a part of the lab works on AMD, but another part works on monogenic diseases like Best disease, and we collaborate with other laboratories for diseases like albinism and ciliopathy. So we make RPE cells from these patients and really try to understand how these diseases, which are known to affect the eye and likely affect the RPE, how they actually happen. Because usually these patients, when they go to a clinician, it's end stage, when the vision has already been affected. At that stage, you cannot tell how the disease actually started 5 years ago when patient didn't even realize. So that's, to me, is the biggest power of this technology, and that's what we're trying to harness. First, to study diseases, and also I think I would say there's a big power in iPS cells for transplantation, because this is patient's own material. It will not get rejected when you transplant it back into patients. So anything made from iPS cells would be well tolerated by them. So that's why this technology is very powerful, and we're trying to harness both of those powers.
So are you collaborating with retinal surgeons? Because ultimately, the technical perspective is putting those into the subretinal space, yes, or in whatever location where they have the best chance of implantation and function.
Yes, I think that's a great point. A very good colleague of mine and a mentor told me about 3-4 years ago, when I was trying to make these cells in a dish, and I said, Kapil, you can make the best cells in the world you want. If you cannot put them at the right place, they're useless. So absolutely, none of this is possible without collaboration with the right people. Just to make all of this, we have to use stem cell biologists, developmental biologists, and bioengineers in the lab. And then to transfer it to patients, we need surgeons, we need clinicians. So right now, for all our animal studies, we have a resident surgeon who actually trained as a human surgeon but doesn't practice anymore in human. He's doing all the animal studies, Dr. Juan Amaral, and he's outstanding. He's transplanting these tissues in pigs to see how they work. Going forward in the clinic, we have at NEI Dr. Henry Wiley. He's our resident surgeon who's experienced VR surgeon, does surgeries, and Dr. Steve Charles. He's a surgeon in Tennessee and has graciously agreed to be part of this. So both of them together are going to be transplanting these patches, RP patches, in patients.
So if you look forward to the first cohort of clinical trials, where will these be conducted, and is there a listing on clinicaltrials.org?
So the trial, phase 1 trial, we are so far planning to conduct all of it at NEI. It would be 10 to 12 patients, so we think we can handle all of that at NEI. The manufacturing of cells will also be done at the NIH Clinical Center. We have a GMP facility that can do manufacturing, and it's literally one floor down from the facility where our surgery suite is, so it's a very easy way to transport the cells and do the trial. It's not announced yet because we are in the process of getting IRB approval for our trial and also to file the FDA paperwork for the phase 1 IND. Once that's approved, it will be announced on the clinicaltrials.gov website.
And what types of patients will you be looking for?
Since this is a phase 1 trial, so it's primarily safety trial. So it will be advanced GA, advanced dry AMD patients, likely starting with the first cohort of more than 2,100 visual acuity. And we're hoping in the second or the third cohort we can get down to a slightly better visual acuity so that we can start to see some signs of efficacy. But since it's a phase 1, the primary goal is a safety trial.
Are there any absolute exclusions?
Not necessarily, because, and again, the advantage is, this is an autologous therapy. We don't have to worry about whether patients had a history of cancer or not, because when you do an allogeneic trial, you have to give immunosuppressants. And with that, you worry whether the cancer might come back if you suppress immune cells that are not able to take care of cancer cells. So since we don't have that worry, that's not really an exclusion criteria for us. I think the only criteria might be they might want to go for very clear dry AMD patients in the first cohort, not with a mixed dry or wet or other ocular complications, but not necessarily any real exclusion criteria.
Excellent. Well, this is an extraordinary line of research, and we thank you so much for coming and speaking with us, and we're going to watch very closely and looking forward to some great results in the future.
Thank you very much.
Bharti, thank you so much.
Thank you for having me. My
Thank
pleasure.
you.